Welcome to the Nitrous Oxide station of our self-guided 4-hour field tour. And well, what is nitrous oxide? You've heard quite a bit about nitrous oxide. It's N2O. It's a gas that is produced in soil by the transformation of nitrogen, any kind of nitrogen that would be in fertilizers, in manures, even crop residues added to soil. And it's involved particularly when ammonium in soil becomes nitrified to nitrate. N2O can be released, and then also when nitrate is converted or in effect reduced to nitrogen gas back to the atmosphere, some of it can be released as N2O. And a big thing that's required here for N2O emissions is water, in terms of water in soil, to kind of restrict aeration a bit. And then the microbes, the bacteria particularly, can then utilize the ammonium oxidation producing N2O or nitrate reduction to produce N2O. Now, why is it important? Why do we talk so much about N2O? It's because it's a greenhouse gas, but it's also a gas that we don't talk as much about, but is important, and that is it is a gas that can break down ozone in the upper atmosphere, and that's problematic because it'll cause us greater ultraviolet light incidence at the surface, can cause problems in terms of for plants, animals, humans, in terms of sunburning and other issues in terms of, as you know, about ultraviolet concerns. Now, the other thing about N2O is that it's produced dominantly, or the majority of the gas that's produced by human activity is actually from agriculture, production of food, and that's because we need nitrogen, and we need accelerated nitrogen cycling in our agronomic systems to produce lush biomass like we're seeing here, and high yield in grain and biomass that we may harvest. So we need nitrogen. It can be added as manures, it can be added as fertilizers, crop residues, organic sources, but some of it is going to be released as N2O. Now, what does this relate to 4Rs? Our research here at the University of Manitoba in Manitoba, but then also around the world with colleagues in Canada and elsewhere, is really showing how using the 4Rs of the right rate, source, placement, and timing can really affect how much N2O is produced, but also when it is produced. Is it early, is it late, or is it not at all even produced during the growing season or even the shoulder seasons in Manitoba, which would be the fall and the spring. Not much happens in the winter because our soils tend to freeze and arrest activity, unlike some other places that soils don't freeze in the winter. Now, the project, in terms of the 4Rs, is looking at 3 project groups: pre-plant and at-plant broadcast incorporation of urea-based sources and their impacts on N2O. Second group being in-season top dressing of UAN with or without inhibitors, particularly urease and nitrification inhibitors. And the third treatment group is looking at in-season UAN placement through surface dribbling, bands of UAN, or side dressing at a shallow or deep placement. What have we found for the past 2 years so far? This is our 3rd year. Well, pre or at planting additions, we found that ESN urea mixture blend can reduce N2O emissions, and we think it's because of the ESN, the polymer coated urea, slows down the release of the urea in the granule to the soil. So it doesn't come out as one big jolt, but it's slowly released. And that is related with, I think, greater opportunity for uptake of that nitrogen by microbes and even soil, through maybe perhaps fixation in some cases, clay fixation, but also through plant uptake. We're studying in the laboratory through process-oriented studies jar incubation type studies what the impact of this slow release is actually on N2O kinetics and dynamics of the microbes and how fast they're producing N2O. And it seems to slow it down in terms of the N2O that's produced. So that's pretty neat. The other is that inclusion of nitrification inhibitor, and in this study, in particular in terms of we're measuring, is with DCD, which is nitrification inhibitor, dicyanamide, and it's in the product Super U, which is one of the two inhibitors in Super U. Super U has a urease and a nitrification inhibitor, DCD, and that seems to slow down nitrification and much reduces N2O. Both of those products, the ESN blend of urea and the Super U, reduce N2O by about 50%. And we've seen this with other types of nitrification inhibitors as well, in trench, which is a different, which is nitropyrin in soil, and we've actually in other field trials with DMPSA, another product that we're getting about 30 to 50% consistently in terms of N2O reduction. So pretty significant reduction. So there's hope in reducing N2O by selecting the source of your fertilizer. Now, in terms of in-season application, N2O seems to be higher for in-season application than at planting. Now, that could be just because of the 2 years that we've done this research at, and in terms of weather dynamics, in terms of soil moisture and rain patterns. But what we've seen is the in-season application is producing more N2O. That may be, I think, this quite related to the temperatures being higher at that time, and therefore the nitrification being a lot more rapid, gives it more opportunity for N2O emission, especially after a little bit of a rainfall that can limit the amount of oxygen in the soil and cause that N2O to be emitted during nitrification. Now, inclusion of the nitrification inhibitor, the DCD, with UAN as a top dress with the product Agrotane Plus reduced N2O by 2/3. So that's pretty amazing actually. So that in season, after the V4 stage, when we see spike of N2O, the Agrotane Plus reduced it quite a bit. Now the surface dribble banded produced more N2O than side dressing. This kind of makes sense because the product, the UAN, is at the surface and it's closer to the atmosphere, and doesn't have a chance to be altered, the N2O, and can get to the atmosphere. And there seemed to be a slight reduction in N2O with deeper side dressings. The deeper is about 3 inches compared to 1 inch side dressing. Not as much of a reduction as I thought would be happening. So I was curious I thought that the N2O would be quite reduced with deeper, because the opportunity of the soil over those 3 inches to the surface trapping and converting and consuming that N2O doesn't seem to be happening. Maybe it's because we needed more moisture maybe in the soil at that time. They've been fairly dry mid-seasons in 2018 and 2019. So we'll have to see what 2020 has in store for us. Can't predict things, as you know. Okay, thank you very much.